We know that DNA is the genetic material, the hereditary material of all life, and it has all this information inside it. But how do our bodies actually use that? How do we change that into traits that we can actually see or use in our bodies? Well, we have to go from DNA and change it into mRNA and then make proteins, which are useful for making enzymes or the structures in your cells and a whole bunch of other things. So first of all, we'll look at prokaryotes and then we'll look at eukaryotes. So this first step of changing DNA into mRNA, we call that transcription because it's still within the same language of nucleotides, still just a different type of nucleotides from DNA to RNA, but then when we're changing it from RNA to protein, we call that translation because you're changing the language. Here it's nucleotides and here it's going to be amino acids. So let's start off with our prokaryotes. So the enzyme that we're going to be using is called Rna Polymerase. So this is like the core dark blue enzyme you see over here. And then we're also going to need a sigma factor, which is a smaller part that's going to bind to this Rna polymerase. And together that's going to make a holo enzyme. Now there's a bunch of different types of sigma factors. Just keep that in mind. It's not just the one thing. Now when we're starting transcription, we're going to start with initiation. So this sigma factor is what's going to help find the specific part of the gene, of the DNA that we want to transcribe. So say this is our gene over here in pink and we want to use this to eventually make protein. Well, first of all, how are we going to find it? There's this thing called this promoter sequence that's going to be a bit before. So this promoter sequence has two key parts on it. We call that the -35 box and the -10 box. And that's just like these specific areas that the sigma factor is going to see, and it's going to use them to orient the RNA polymerase in which way it wants to be going down the DNA, right? So now we're going to, the sigma factor again is going to help open this DNA double helix so that we can have just the individual parts. Now the RNA polymerase can actually start transcribing. So notice that the top part over here, which we have from 5' to 3', this is what we call the coding strand or the non-template strand. So this has the gene, this has the promoter sequence or promoter region, but that is not the one that we are going to be using to actually make the RNA. So the RNA is going to be synthesized using the template strand because it's a template for making RNA. So this strand is going from 3' to 5' because when we're adding nucleotides to it, remember they're nucleosides triphosphates, which will then become monophosphates when you add them on. Now these have to be added from 5' to 3'. So for RNA polymerase, it's going to be moving down this way and adding those nucleotides 5' to 3', forming this mRNA. So as soon as we get the sigma factor is going to help the RNA polymerase, it's going to find this promoter sequence, it's going to open up the DNA, and then we're going to start transcription. Once transcription is started, well, the sigma factor is like, okay, I'm done my job. It's going to leave, and then the RNA polymerase is going to continue this elongation. After that, when we want to finish, when we're done transcribing, we're going to end it, so we call that termination. So what's going to happen is there's the sequence actually in the DNA that's called the termination sequence, and that's pretty much just if you have a few like nucleotides that will pair up or complementary base pair with other nucleotides that you're creating later on, like just a short distance away, but they're going to actually complementary base pair in this RNA, even though it is single-stranded, right? But it's going to form this kind of hairpin loop structure over here, and that is going to cause this RNA polymerase to stop transcription. It's going to dissociate and leave the DNA by itself, and then the newly formed RNA can now be used later on for translation. And now that we're done that part, the RNA polymerase can go back and rebind with sigma factor, and we can continue with new genes to make more proteins. So transcription in eukaryotes is a bit more complex First of all, keep in mind this is happening in cytosol, and this is happening in the nucleus. So DNA in eukaryotes is a lot more tightly packed around all these histones and stuff, so it's harder to get to in the first place. And then when you do get to it, it's not just as simple as having a core enzyme like your RNA polymerase and then different types of sigma factors. For this one, you actually need there's different types of RNA polymerases, first of all. So RNA polymerase 2 is going to be your focus because that's the one that's going to actually make proteins, like mRNA that's going to be used for proteins. But there are other types of RNA polymerases. Won't talk too much about those. And then there's also all these other transcription factors that are going to help the RNA polymerase during the process of transcription. So first of all, here we're going to have the initiation. It's going to start by this TATA-binding protein, which is going to find this region that we call the TATA box. So instead of having promoter sequences like we did in initiation of prokaryotes, where you had the 35, the minus 35 and the minus 10 boxes, we have this thing called a TATA box. A TATA box is just like this region of thymines and adenines, and that is where we're going to start, like the TATA binding protein is going to bind, and it's kind of, so if this is my DNA, and this is our TATA binding protein, it's going to move along, and it's going to start bending the DNA so that we can read it better, and once it finds this spot over here, it's going to bind to it, and then you can have all your little RNA over here, polymerase, with all these other factors helping it. Normally they wouldn't fall off, but yeah, so they'd be coming along and they would continue to move down the DNA so that we can now transcribe our gene. So at the end, after that, once you start transcription, all these little guys, the other factors, they're not needed anymore, so they're going to dissociate and leave. And then there's going to be these other factors at the end that are going to say, okay, we're done. The mRNA will be separated, and again, the RNA polymerase is going to dissociate and come back and remake this complex so that we can continue with more transcription with other genes.